All right, we end up our metabolism unit by taking a look at what's going on in the electron transport chain during aerobic cellular respiration. And just like, this probably looks familiar to you because it looks similar to what's going on in the thylakoid membrane during the light-dependent reactions of photosynthesis. And it's a very similar process. So let's start by identifying everything. Let's start with the inner membrane. And the inner membrane is a phospholipid bilayer, just like all biological membranes. And then what gives it its function are the proteins that are embedded in it. So let's go ahead and identify those. The first one here is a proton pump. So it's pumping hydrogen ions across the inner membrane. And notice that it's pumping it from the matrix, in other words, inside the inner membrane, to the intermembrane space, out into the intermembrane space. So that means we're building up hydrogen ions in the intermembrane space. And remember, that would decrease the pH, because more hydrogen ion means more acidic, right? There's that inverse relationship. Another proton pump. Another proton pump. So we have, unlike in the thylakoid membrane and during photosynthesis, the light-dependent reactions, there was only one proton pump then. Here we have 3 proton pumps. So as these electrons are traveling through the electron transport chain here, they are pumping a lot of hydrogen ions across the inner membrane and out into the intermembrane space. These are electron shuttles, just like I told you they were in the electron transport chain of the light-dependent reactions. But here they're referred to as cytochromes. We're going to call them cytochromes. Cytochromes are responsible for shuttling electrons in between the proton pumps, basically, and completing the pathway for electrons to travel through the electron transport chain. And then the last protein that we have embedded in the membrane is ATP synthase. The same ATP synthase that we saw in the light-dependent reactions of photosynthesis embedded in the thylakoid membrane. All right, so let's take a look at what's going on when electrons are delivered and pass through the electron transport chain. Remember, these electrons are coming from glycolysis, the transition reaction, and the Krebs cycle, and they're being delivered by electron carriers, NADH and FADH2. And notice that there are 2 electrons that are being dropped off by each of these, and so they travel through the electron transport chain in pairs. And for every 2 electrons that are dropped off, on average, there are 3 molecules of ATP made. And you might remember that because it kind of goes along with the 3 proton pumps. So for each pair of electrons that passes through a proton pump, an ATP will be made. But that's on average. And the discrepancy in that is that notice that FADH2 is being dropped off in a different place. It's being dropped off after the first proton pump, which means the electrons that it's dropping off aren't going to be used to pump as many protons. So the electrons pass through the first proton pump, get picked up by cytochrome, delivered to the second proton pump, picked up by cytochrome, delivered to the third proton pump, and by the time they energize all this pumping of hydrogen ions across the membrane, or protons across the membrane, they've lost energy. And they're going to be picked up then and combined with hydrogen and oxygen to make water. And we refer to this as metabolic water because it's being produced by metabolism. So that means that all the mitochondria and all the cells of all your body, of your whole body, in carrying out aerobic cellular respiration are making water. Unfortunately, it's not enough water that you don't have to drink. So you're losing more water than is made through metabolism. However, there are some animals that are able to survive on just the water made through their metabolism and they get from the food, because food contains water, but they don't have to drink. So there's a kangaroo rat that lives in the desert that doesn't have to drink because of its metabolic water and the water it gets from its food. But the point here is that oxygen is necessary as the final electron acceptor at the end of the electron transport chain. And if oxygen is not here to pick up those electrons, the electron transport chain stops. And if the electron transport chain stops, the Krebs cycle stops. And if the Krebs cycle stops, the transition reaction stops. So everything that goes on within the mitochondria relies on oxygen being here at the end of the electron transport chain. And that's why you're breathing in oxygen. And that's what makes this process aerobic. So in the meantime, we've pumped all these protons across the membrane, build up this high concentration of hydrogen ions in the intermembrane space, and a relatively low concentration of hydrogen ions in the matrix. So what we have, of course, is a concentration gradient, and that's potential energy. Having a concentration gradient is potential energy, and the name of that has been given the proton motive force. So this has built up a proton motive force, and that energy can be used to drive ATP synthase. So again, as I pointed out when we were looking at the light-dependent reactions of photosynthesis, the way ATP synthase operates is by physically, mechanically turning. And they've shown this experimentally, that it actually does turn. And it's the motion, that mechanical motion, the energy of that mechanical motion that's used to add a phosphate onto ADP to make ATP. And that's inorganic phosphate. So as opposed to previously when we've seen ATP being made during glycolysis and the Krebs cycle, if you'll recall, that's by substrate level phosphorylation, because the phosphate's coming off of an organic substrate and placed onto ADP to make ATP. But here, it's inorganic phosphate, and because it involves oxygen and oxidation of glucose overall, we refer to this as oxidative phosphorylation. So you now know of 3 different kinds of phosphorylation. There's photophosphorylation that's taking place during the light-dependent reactions of photosynthesis. There's substrate-level phosphorylation that takes place during glycolysis and the Krebs cycle. And here we have oxidative phosphorylation, phosphorylation of ADP to make ATP. And then the other thing to note here is that this process, as it was in the light-dependent reactions of photosynthesis, is known as chemiosmosis. The diffusion of hydrogen ions through ATP synthase to make ATP is referred to as chemiosmosis. So NADH from glucose, from glycolysis, the linking reaction, and the Krebs cycle, that's where NADH comes from, and FADH comes only from the Krebs cycle, if you'll recall. But they have different entry points in the electron transport chain, so they're delivering different energies to pump protons. And as always, all of these reactions and actions are regulated by enzymes. So we're not dealing with any of the enzymes except for ATP synthase. Remember, that's an enzyme. But there are enzymes that are regulating all these other reactions that are taking place and actions too. Enzymes run everything. So here we're going to tally up all the energy we get from one glucose molecule in all these different parts of aerobic cellular respiration. And just remember, as I pointed out on the previous slide, on average, about 3 molecules of ATP are produced for each pair of electrons that travel through the electron transport chain. So just to add some things to the diagram to make it more understandable, we've got the transition reaction going on here. This is glycolysis, right? This area here has been shaded to show it's glycolysis. But they don't point out the transition reaction, so that's what this is representing. They did box this whole thing off as being the Krebs cycle and the electron transport chain. So this is the transition reaction. This is the acetyl group that CoA binds to help it get into the Krebs cycle. So that's, again, the transition reaction. These electrons, anywhere in this diagram there's NADH or FADH2, those are electrons that are going to be delivered to the electron transport chain. And that's going to, just like we were looking at on the previous slide, those electrons are going to participate in oxidative phosphorylation by chemiosmosis, the production of ATP by oxidative phosphorylation and chemiosmosis. or by chemiosmosis and oxidative phosphorylation, however you want to say it. So there we have NADH, here we have NADH, here we have NADH, and here we have FADH2, and they're all delivering, or funneling, as we say, those electrons to the electron transport chain. Also, to clarify the diagram and relate it to what we've been learning, the ATPs are being produced in different ways, right? So like from glycolysis, that's substrate level phosphorylation. The phosphate's coming from a substrate, one of the intermediate substrates during glycolysis. Same for the Krebs cycle, substrate level phosphorylation. But then from the electron transport chain, that's oxidative phosphorylation, right? That's already labeled up here. So glucose has 6 carbons, and all 6 carbons are being stripped off and glucose is being broken down, and that means stripping off carbons and harvesting electrons. And every time a carbon is stripped off, that releases carbon dioxide. And there's 2 places where that's happening. There are 2 carbons that come from each glucose molecule, 2 carbons from the transition reaction, and then there are 4 carbons from the Krebs cycle. So that's the 6 carbons from glucose. As far as efficiency goes, aerobic cellular respiration is about 18 times as efficient as anaerobic fermentation. Because remember, the fermenters are only getting these 2 ATP from glycolysis. That's all they get. But remember, there is still an advantage because it doesn't require oxygen and it's very fast. So as long as there's plenty of glucose, it's an efficient way to get a lot of ATP. But aerobic cellular respiration is about 40% efficient, which is pretty good. I mean, the best internal combustion engines, in other words, cars, gasoline engines and diesel engines in cars, are barely approaching 40%. They're more like 25, 30%, I think. So aerobic cellular respiration is pretty efficient. And that efficiency rating goes along with that analogy that I mentioned to you before, where glucose, if it were a $100 bill, ATP would be $1 bills. And so we have our total of 38 ATP, and that's about 40%. 38 out of 100 is 38%. That's almost 40%. So that analogy fits, and it can help you remember the efficiency of the process. So if it's only 40% efficient, where does the rest of the energy go? Well, it's lost as heat, and that heat contributes to the homeostasis of body temperature in us. Other organisms that don't maintain their body temperature, it's just dissipated from their body. But that's where it goes. So the total that I've given you is 38 ATP from one glucose molecule. The book says 36. Now, I'm going to explain the discrepancy between those two numbers. You don't need to know this. I'm not going to ask you to reproduce this on a test or a quiz or anything. But what it has to do with is the way that the NADH that's produced during glycolysis is shuttled across the outer and inner membranes of the mitochondria to get it into the matrix to participate in the electron transport chain, or to get it into the inner membrane where it participates in the electron transport chain. So they're picked up in the inner membrane space by either NADH or FADH2, and that's the key. Remember, FADH2 enters the electron transport chain after the first proton pump, so there are fewer protons being pumped across the membrane to make ATP. So this NADH is going to participate in the electron transport chain as either NADH or FADH2 because of the way it's shuttled across the membrane. So that's the reason for the discrepancy. So what I'm giving you is the total possible maximum of 38. That's if the NADH from glycolysis is shuttled across the membrane as NADH and doesn't get swapped over to FADH2. But again, I just wanted to let you know that that's where the discrepancy comes from. So we end up now with taking a look at that ecological relationship between the two processes and then running through some of the important vocabulary that goes along with this. So one last time, the products of one are the reactants of the other. We're looking at photosynthesis and aerobic cellular respiration here, the overall reactions. The products of one are the reactants of the other. Products of one are the reactants of the other. Matter cycles and energy flows. So these arrows represent the cycling of matter, but the energy is going into photosynthesis and coming out of aerobic cellular respiration, and that's a one-way street. That energy that goes into photosynthesis and into glucose then is broken down during aerobic cellular respiration and released. It doesn't cycle, right? It just flows straight through from one process to the next. And that energy is then going to be captured as ATP, and that ATP then is what's going to be used for cellular work to power life. Some of the terminology that hopefully you remember is related to the whole thing. The producers are doing this, and this could be chemosynthesis too, so don't forget about chemosynthesis. So producers, consumers are carrying out aerobic cellular respiration, but remember that the producers also, even though they're making their own food, they're making glucose, they still need to break it down to get the energy back out. They're making that glucose as a way to store energy, and they need to break it down and get it out just like consumers do. So, and producers here, I just put it a little aside, but you need to remember that producers have mitochondria too, and they're breaking down the glucose that they make for themselves. So those cell feeders, right, are autotrophs, and they can be either chemoautotrophs or photoautotrophs. We're looking at photoautotroph, photosynthesis here. But that's an endergonic reaction, right? That term means it's an energy-absorbing reaction. Energy has to go into the reaction. Heterotrophs, in general, are carrying out aerobic cellular respiration, but don't forget that autotrophs are also carrying out aerobic cellular respiration too. They have to break down the food that they make, and aerobic cellular respiration is an exergonic reaction. Energy comes out, right? So that term goes along with what's going on in aerobic cellular respiration. And then remember, there are two halves of metabolism. So overall, photosynthesis can be considered an anabolic process because we're building glucose. And aerobic cellular respiration can be considered a catabolic process because it breaks down glucose. So building up, breaking down. Two different kinds of reactions that we find in metabolism. That's it. And this is a huge unit, as you know, I'm sure you've realized. And that's why you have this helper sheet project where you are recording all these details, both the ecological aspects of metabolism and photosynthesis metabolism and aerobic cellular respiration metabolism. And I don't expect you to remember all these little details. I expect you to have them on your project, and then you can use your project to be able to score high on the upcoming test. So good luck with that, and get to work.